Hello guys, I am Harun and it's your YouTube channel Harun biozone. Today's in this video I will discuss about the structure of a double-stranded DNA or double helical structure of DNA. Double helical structure of DNA by Watson and Crick 1953. So first, let us look. Watson and Crick first described the structure of the DNA double helix in 1953 using the X-ray diffraction data of DNA fibers obtained by Rosalind Franklin and Wilkins. M. Wilkins. The structure of DNA is represented by or produced made by Watson and Crick and R. Franklin, Rosalind Franklin, M. Wilkins. Watson, Crick, and Wilkins are awarded 1962 by Nobel Prize, and for this they take a Nobel Prize in 1962. Except Rosalind Franklin. She is called Black Lady of the DNA. Black Lady of the DNA because in that time the dead person do not get the Nobel Prize, but nowadays dead person also get Nobel Prize on the basis of his work. So Rosalind Franklin died before 1962, so she did not get the Nobel Prize, but Nobel Prize get by Watson, Crick, and Wilkins. This is the structure of characteristic feature of the structure of DNA. Two long polynucleotide strands coiled around a central axis. Suppose this is the central axis and two polynucleotide chains are coiled around the axis. Clear? What is polynucleotide? What is nucleotide? Nucleotide is a structure that contain ribose sugar, 5-carbon deoxyribose sugar, and nitrogen base and phosphate group. Okay, and many nucleotide joined by phosphodiester bond. So phosphate many nucleotide joined by phosphodiester bond to produce a polynucleotide chain. Number 2, strands are wrapped plectronomically in a right-handed helix. Around this axis, the strands are right-handedly wrapped. Number 3, strands are antiparallel because one 5' to 3' and another is 3' to 5'. Why antiparallel? Because their stability, increase their stability. Number 3, strand interact by hydrogen bond between complementary base pair, that is adenine always combined with thymine by 2 hydrogen bond and guanine always combined with cytosine with 3 hydrogen bond. Angles interaction between base pair result in major and minor groups. This groups is called major groups, and this is called the minor groups. Number helix diameter 20 angstrom. If we imagine it is the axis and here this 10 angstrom and this side also 10 angstrom. The total diameter of the DNA is 20 angstrom. Helix per rise per base pair. Here the correct structure of DNA. This is a 2 strand of DNA helix and this is a central axis. Okay, this is a central axis. Here 5' to 3' and 3' to 5' to 3 prime antiparallel structure. This is called minor groups, this is called major groups. Okay, and and these major groups and minor groups made a complete turn. The complete turn, okay, in this complete turn, total 10.4 base pair per turn, and the distance between the pitch or distance between two base pairs after adding thymine and guanine and cytosine. So the distance or pitch between two base pair is 3.4 angstrom. Okay, so total number of base pair is 10 base pair in a complete turn, in a complete 10 base pair in a complete turn. So 10 into 3.4, that is 34 angstrom, is the size of a complete turn, where one major groups and one minor groups are present. Then helix rise per base pair 3.32, but accurate is 3.34. Okay, 3.32 or 3.34, no problem. Helix pitches distance along the axis per 360-degree turn 33.2 angstrom. 33.2 angstrom means it's 34 angstrom. This turn, helix turn. 10.4 base pair per helical. 10.4 base pair per helicals are present. Per helicals means per complete turn present 10.4 base pair or 10 base pair of DNA, 10 base pair of nitrogen base. Base pair are in the inside of the molecule stacked close to each other, and base pair are stacked close together by hydrogen bond. So what is the common features? The diameter of the DNA is 20 angstrom. 20 angstrom. The distance between two nitrogen base pair 3.4 angstrom, okay, or 0.34 nanometer. One complete turn contains one major groups, one minor groups, and here is 10 base pair DNA are present. So 10 base pair 3.4, that is 34 angstrom or 3.4 nanometer is the distance between complete turn. Okay, that DNA strand is anti-parallel. The one strand is 5.3 5 to 3 5 prime to 3 prime and 3 prime to 5 prime. Why anti-parallel? Because due to their stability. Number 4, next, stability, and this double helical structure maintain the Chargaff's rules. Chargaff's rules, that is adenine plus guanine, purine by pyrimidine on, or adenine plus guanine is cytosine plus thymine, or adenine always bind with thymine by 2 hydrogen bond, guanine always bind with cytosine by 3 hydrogen bond. Okay, this is the structure of DNA helix. Thank you.